Hello students and welcome back to my channel. This is the Toxicology series of videos and today we're going to continue by talking about a few things. Expect to learn in this lesson the etiology of toxicology, the various factors that modify the toxicity, as well as the methodology we use to evaluate the toxicology. Hello and welcome back. My name is Tommy. I'm a human nutrition student and researcher at the university. What I do in this type of videos is to synthesize what I learned at the university, so then I can explain and teach you the best and most valuable information you need to know. So let's begin this video with the etiology of toxicology. So we have harmful toxins here that attack living organisms, and we ask ourselves, what is this doing? So they create diseases. Disease caused by toxicokinetics and toxicodynamics. So quantity and speed of toxins is very, very important. These can create diseases. Indeed, the dose is the major factor here. Remember Paracelsus. Legislation. So it's different when we talk about intoxication versus poisoning. The intentionality differs because poisoning is with an intention. And then there is also the drug addiction, which is self-induced. There are different types of the intoxication. By level, it can be light, mild, or severe. It can be immediate or with delayed effects, reversible or not, local or systemic. If we take into account the time, it can be acute, so the symptoms and effects are present in less than 24 hours and with only one dose. Subacute, so the symptoms within a few days. Chronic, which is a repetitive intoxication or repetitive exposure to this toxin at lower doses. So this is very difficult to detect, but it appears to be the most frequent one. And the recurrent type. So it is a recurrence of clinical signs. You feel better for a bit and then you get the signs again, signs and symptoms. If we take a look at intentionally intoxication, we have poisoning, such as assassination or even abortion, hedonistic, so social drugs and doping for sports, and chemical and biological weapons, such as the ones that they used in world wars. And then accidental intoxication can be either environmental, so acid rain and chemicals, occupational, so remember Bernardino Ramazzini, lower hygiene of the workers, and the environment polluted with chemicals. Drugs, medicinal drugs, errors in type or dose prescription, food, natural toxins, or even food containers can create an intoxication, BPA for example. Microorganisms. There is a difference between intoxication, when we ingest the toxins, or toxinfection, when we ingest the microorganism with the toxin. Domestic, we have children mainly, but also the elderly. This is, for example, when they drink some cleaning products, thinking they are energy drinks or something like that. And then biotoxins. These are created by living organisms. So all of these are the types of intoxication we can have. But fortunately for us, we have antitoxin centers, and they are specialized in information and advice services, as well as analysis and treatment. So now let's take a look at the factors that modify the toxicity. We have environmental factors first. So the weather. Yeah, the weather changes our mood and also our response to toxins. Atmospheric ions and noise will act on our central nervous system, making us weaker. Then also light. We have the actinism, where light increases toxicity, and also the saturnism, which is a lead poisoning, and this disease appears to be higher in the summer, so when there is a lot of light exposure. And indeed the sun can increase our sensitivity to the toxins. Temperature, so higher body temperature will increase the chemical reactions speed and will increase our defense against the toxins. Indeed, the temperature affects body functions, toxicokinetics and toxicodynamics. So the absorption, distribution, metabolism and elimination of these toxins. And the majority of drugs have the least toxicity at medium temperature, following a U type. Some of them are linear and others maintain the level to then increase. And then also pressure. Pressure affects toxicodynamic and the circadian rhythm. Higher pressure, higher toxicity. Individual factors, so the species. In front of the same drug, different species have different toxicity levels. And we use rats, monkeys, and pigs because they are similar to humans. We use them to try the toxins to then determine if they are toxic for us as well. The race. Blacks have more defense mechanism than whites or Asians, for example. And in sex, different hormones, different response to toxins. There are some toxins that affect more men than women and vice versa. The age. So kids are most sensitive to drugs, and the elderly as well. They reach toxicokinetics saturation faster than the adults. Indeed, the geriatric age, they have a lower function of the liver and kidney, a higher pH of the gastric juice, so lower projection and digestion, lower intestinal motility together with a mucosal atrophy, lower plasma albumin and other transport proteins, higher adipose tissue and less muscle mass, as well as total body water. And then there is also this factor, which is the idiosyncrasy. These are characteristics that distinguish an individual from another. We are all different. Even individual twins can be different. And here we differentiate between hypo and hypersensitivity, following the Gauss bell. So hyposensitivity and hypersensitivity are related to allergies, for example. Hemoglobins H, M, or S can lead to the formation of metahemoglobin. Favism, a deficit in the glucose-6-phosphate dehydrogenase, will lead to an hemolytic anemia with genetic character. And also, the acetyltransferase sensitivity is a type of allergy related to hypo or hypersensitivity. Toxic resistance varies by resistant mutant selection, the enzymatic mutations that appear, biochemical modifications, the tolerance we have to the toxin, disease, so organ deficits, fatigue, and the psychosocial state as well will influence our resistance to toxins. If we feel lonely, we will have a lower immune system. Same with drug addiction and suicide behavior. And now let's talk about the methodology for toxicological evaluation. So there are two main principles here. The first one is that animals' results are applicable to humans, as we talked about before. There's a similarity between humans and animals' limits. And there is a factor of 10. We use this factor to make the conversion between animals and humans. We use mice, pigs, and primates because they are very similar to us. And the body weight and surface area are the main factors to take in account. The second principle is that animals' doses must be high in order to properly calculate their population average. This is the dose-response concept, in which the majority of the population at that dose will have the effects. Dose-effect relation, harmful to humans or the environment. And these are the dangerous health effects: lethal acute effects, irreversible non-lethal effects, serious effects, such as repeated or prolonged exposure. Very, very dangerous. Then we also have corrosive effects, irritant, sensitizing, carcinogenic, mutagenic effects, toxic effects on reproduction and development, and also effects on the environment. So now let's view from where we gather the knowledge of the toxicity of substances. We have theoretical predictions, retrospective studies of intoxication, experimental tests with animals and plants, and human studies. These human studies are retrospective, epidemiological, such as these ones, or volunteer trials. Let's take a look at the experimental tests with animals and plants, starting with plants. Knowledge of the phytotoxicity of agricultural products and phytosanitary products. So here we have to take in account all those phytosanitary products and agricultural products such as DDT or other pesticides we use in plants. And we also need the knowledge of the metabolism, transformation and retention of toxic substances by vegetables. So we need to know how the metabolism of the plant will work on those pesticides, how many of those are going to be stored in the plant, and if it is a potential danger for us when we eat them. An example of these experimentations with plants is the cultures of unicellular algae that can detect herbicides in food. Very interesting. Experimentation with amphibians. So we have two methods here. The main one is the race method, and it is the study of mycotoxins. We use larvae of amphibians, tadpoles, and inhibition of egg and larval development. The other method is the Russell and Birch, which is the 3Rs method. It focuses on reduction, refinement, and replacement, with the goal of lowering the waste, so to use as less animals as possible. And then experimentation with animals. Traditional protocol of experimental toxicology. We have to choose the animal species. There are different types of animals, of course. We recommend small animals because they are economic, and big animals only in certain studies. Transgenic animals only in certain studies as well. We have to choose 50% of both sexes and multiple species to avoid biases. And then the animal testing. So if we test oral or parenteral toxins, we use mice and dogs. If it is a topic substance, rabbit. And sensibility test, guinea pig. Ways of administration to use. It depends on the type of and absorption place in humans. So we use a control group of study with animals, and the toxicity is different depending on the route of administration, of course. And we can have oral, parenteral, and dermal routes. There are tests for skin and eye irritants and sensitizers. So then we have to determine the total number and group of animals. So it depends on the homogeneous or heterogeneous result, following the Gauss bell. Again, very important. We use statistics to reduce the amount of animals, so less waste. And then if we take a look at time, we have acute and chronic. Acute, we give only one dose and 24 hours to see the effects. There is an estimation of the quantitative toxicity and identification of the target organs. Very important. And we have to select the doses, of course. What are the methods? The up and down, so with 4 animals, trial and error. The log method with 3 animals of one sex, oral toxicity class, and fixed dose method, using the parenteral way of administration. And the goal is to find the lethal dose 50, which is the dose that's lethal in the 50% of the cases. So that will kill 50% of the population, 50% of the group. It varies a lot in different ways of administration and animal species. Chronic studies, they are done throughout life. And it's better to choose mice over a tortoise, because mice live for 2 years, more or less, and tortoises for hundreds of years. So for convenience, we will use mice. Duration of administration and observation and analysis of the animals. We have to considerate the longevity weight. So again, the weight is very important here. These are 6 to 18 months studies, 90 days histological study, and the daily administration of the toxin, because they are chronic. They can be medium-term studies when they finish after 3 months, or long-term studies over 6 months. And then we have the toxicology of reproduction and development. So in fertility toxicology, there is a lower in libido, physiological responses, lower fertilization, and lower hormones because of those toxins. So less probability of getting pregnant and the impotence in men. Development toxicology, on the other hand, is the defect on growth, functional, structural, and mental growth of the infant, of the fetus, mental including a lower IQ level, possibly. We will take a look at this in more detail in the next video. Study of animal behavior is the last step. There is a selection of the animals, isolation to prevent any visual, auditory, olfactory stimulation. A controlled environment is necessary, and the reduction of feeding. The goal of this is to get objective parameters for the evaluation. The easiest method to study the animal behavior is the observation before and after the toxin administration. Alternative methods will include in vivo, in vitro, or combined methods, and also molecular toxicology, for example, the use of transgenics. Taking a look at comparative toxicology, Litchfield says that if an effect appears in rats and dogs, it will appear in humans as well. So there is a correlation between animals and humans, especially mice and dogs. Kravosky said that there is a correlation between the lethal dose 50 and the body weight. So it's very important, the body weight. And Edwards et al. Said that the calculated doses actually match the intoxications, the real intoxications. So these calculations are accurate. Okay, this is it for this video. Thank you so much for watching. I hope you understood everything I said. If you didn't, there is a comment section down below. Don't forget to like, subscribe, and share this video to your friends and family, to spread this knowledge to them as well. Thank you so much for watching, and I will see you in the next one, in which we're going to talk about the mutagenesis, the teratogenesis and carcinogenesis, as well as antidotes and antagonists, and the risk assessment. I will see you there.